Well, dear students, today we will continue the topic started on the previous lecture on protein biosynthesis, and today we will be talk about translation and post-synthetic modification of polypeptide chains, and about any hereditary diseases associated with its processes. No basic idea which describe all our properties. It transfer of information about functional molecules, main functional molecules in proteins, and information about structure of primary structure of proteins localized in DNA, RNA, intermediate carrier of information, and polypeptide chain of proteins realized by translation. We told that information in DNA localized in any one of double strands of DNA, a template strand, and the orientation and localization of genes in long strands of DNA can be on both strands, but orientation will be on opposite directions because strands organize in opposite directions. One strand is template, other will be coding, and upstream of coding sequence will be localized special place, special sequence of nucleotides, so-called promoter, and more or less long regulative parts, which also in eukaryotes it enhancers and other regulative elements, and affinity and activity of RNA polymerase to DNA, to promoter, and activity of regulative factors lead to different variants of different number of copies of messenger RNA, which will be realized in transcription, and different number of copies which will be translated from this messenger RNA. And a multiplicity of non-coding, intra-coding, non-coding, intra-genetic sequence of DNA and messenger RNA, and different number of introns can lead to multiple variants of transcribated and later translated polypeptide chains. We told that number of polypeptide chains coded by genome, number of genes in human genome at near 21,500 genes, but number of polypeptide chains which coded it genes it near 150,000 polypeptides, and it one of reasons it so-called alternative splicing when in different tissues, in cells in different tissues and organs can realize different variants of splicing with forming of functional active polypeptides. And we finished last lecture by appearance of functional active messenger RNAs, and for example, in prokaryotic organization of genome, near half of all genes can be organized in operonal type when one promoter will be will be lead to synthesis of multiple cistrons, and each cistron consists information about individual polypeptide chain and total functional properties of its polypeptides, its participation in the same metabolic pathway. For example, synthesis of any amino acid, for example, tryptophan or histidine operons, or metabolism of any source of energy, for example, lactose operon, or galactose, or ribinose and other operons. Not all genetic organization in prokaryotes consist operonal type, near half. Other genes have different organization, but it's important for understanding of regulative events in prokaryotes. Eukaryotic messenger RNA organize more complex. It after splicing of exons take place additional modifications. It capping, for example, on 5' end methylguanosine cap. A relatively long 5' untranslated region, which will be consist any specific sequence of interactions with ribosomes, and relatively long also untranslated region, 3' untranslated region, with terminal loop, with other regulative elements and polyadenine tails. And after appearance of messenger RNA will be take place events of translation, synthesis of polypeptide chain, which consists different stages. It activation of amino acids, it really translation steps of translation, initiation, elongation, termination, and the post-translational modifications of polypeptide chains. Some hundreds molecules, some hundreds, some participants participate in these reactions. No, and first step after appearance of messenger RNA, it activation of amino acids. Now you know that all synthetic reactions in all synthetic reactions used activated monomers, activated substances. For example, synthesis of glycogen, in synthesis of glycogen used not free glucose but UDP glucose. Synthesis, for example, soluble bilirubin, it used not glucuronic acid, free glucuronic acid, but UDP glucuronic acid. The same for synthesis of polypeptide chain used not free amino acids, but activated amino acids, macroergic activated amino acids. It result of interaction of amino acid and ATP. You see, amino acid will be interact with carboxy end of amino acid and forming aminoacyl adenylate. And later, aminoacyl adenylate will be interact with transfer RNA. You know that 20 amino acids in polypeptide chain will be interact with next important participant, it transfer RNA. Transfer RNA, it RNA molecule which consists 5' end and 3' end in linear structure, but it consists relatively high concentration of minor nucleotides. For example, pseudouridine and dihydrouridine, modified usual nitrogen bases, and consist repeated sequences, antiparallel sequences, so-called tandem repeats, which will lead to forming of polynucleotide chain of messenger of transfer RNA to stems, to loops which correspondent lead to so-called three-folial structure with so-called arms, dihydrouridyl arms, pseudouridil arm, and anticodon arm. And all tRNAs consist on 3' end the same sequence, CCA, cytosine, adenine. And ribose of 3' end, free ribose, non-associated with next nucleotide, it place of binding, site of binding of activated amino acid, aminoacyl adenylate. And transfer RNA, it relatively short sequence from 75 to 85 nucleotides, relatively low molecular weight. In three-dimensional space, it three-folial structure folded in L-like letter G-light in Kirillitsa, with possibility of specific properties which important for next stages of translation. Now again, activation of amino acid lead to forming of aminoacyl adenylate, relatively macroreergic substance, and aminoacyl adenylate interact with one of free OH group associated with 3' end ribose, or second or third. It depends on enzymes which can to provide it reaction. Result will be association ribose to carboxy end of amino acid. It provided by special group of enzymes which call aminoacyl tRNA synthetase. It the same number of different synthetase like tRNAs. tRNAs, some more than 20. It not 61 number of tRNAs, some more than 20. It depends on genetic code. And each synthetase recognize own tRNA and own activated amino acid, and it lead to association of correspondent amino acid with 3' end of tRNA. You see on one side of tRNA localized anticodon loop, on other side will be correct amino acid with correct tRNA. It's very effectively work enzymes, and we have two groups of it enzymes, aminoacyl tRNA synthetases, two classes of synthetases. Class first provide catalysis association of amino acid with second hydroxy group of 3' end, and second class attach amino acid, activated amino acid with 3' end hydroxy group. No, it's very conservative sequence, very conservative enzymes with very conservative amino acid sequence, and mutation in this sequence usually lead to impossibility of existence of this organism. One of important size of life of each cell, and correspondence between amino acid and sequence of amino acids and sequence of nucleotides described as genetic code. And now you're going to see example of most frequent variant of genetic code which present in cytoplasm of all eukaryotes. And you see that 4 nucleotides organized in triplets, one amino acid coded by one triplet. It properties of genetic code is triplet organization, and you can to see that number of triplets much more than number of amino acids. Really possible 64 variants, and 61 variant of genetic code of triplets coded amino acids. It leads to so-called degenerative code, when usually more than one triplet coded one amino acid. Only 2 amino acids coded by single triplet. It triplet which coded methionine, AUG, and tryptophan, UGG. Other amino acids you can to see coded by 2 triplets or 4 triplets, and one case, for example, leucine coded 6 triplets. And the degenerative properties of genetic code tell about importance first 2 letters, 2 nucleotides in triplet, third nucleotide usually not important. How it's possible to describe as providing of perfectness of synthesis. It describes so-called wobbling when tRNA with number of tRNAs correspond to amino acids, some more maybe up to 22, 23 different variants of tRNAs. For example, for leucine, important not only maybe change first and second nucleotides, you can to see necessary to have different tRNAs, but in complex of tRNA and messenger RNA in ribosome take place adaptation of interactions without serious destroying of binding of third nucleotide. Triplet, degenerative, unambiguous, and comma-less properties of code, and universal. No, universal, it's relatively universal, because now known near 16 variants of genetic code. All of it variants of little changing of most frequent cytoplasmic genetic code. For example, in our mitochondria, other little different genetic code. Now it's about Wobbly hypothesis. First two bases more important. And now you're going to see variants of different genetic codes. For example, in our mitochondria, tryptophan coded by two triplets. For example, vertebrates. You see UGA also not stop, but tryptophan operon, tryptophan codon. In different variants, you can to see this any fungus, in any other eukaryotic organisms. In mitochondria can be methionine also can coded other variants. Well, and now about next steps of translation. In messenger RNA necessary to have specific start signal, which will be beginning of polypeptide chain synthesis. In prokaryotic messenger RNA, upstream of start will be any purine-rich special sequence, near 10 nucleotides upstream of first triplet. And if you will remind about polycistronic messenger RNA, it can be more than one structural genes, cistrons, and each of it will be consist of an AUG. And upstream sequence which will be indicate about first triplet. For eukaryotes, it also will be the same AUG, first methionine, and the cap and upstream special sequence which will be provide interaction with ribosome, important for beginning of synthesis. But maybe next problem. For example, if we have in messenger RNA two triplets AUG, both of it will be start codon. But if will be first AUG start codon, in this case, second it will be not AUG. It will be next triplets CAU, GCA, and more and more. How it possible to determine what triplet will be true? For it necessary special sequence which will be upstream of coding sequence, coding triplet, so-called Scheiner-Dalgarno sequence. And each sequence will be provide correct interaction of true AUG with start of polypeptide chain synthesis. It will be provide correct interaction with ribosome. Next component of synthesis, participant of translation, it ribosome. What is it? It ribonucleoprotein complex, which consists without messenger RNA divided on two subunits. And all of it consists ribosomal RNAs and some tens polypeptides which constantly associate with ribosome. And you know that ribosome divide on 70S ribosomes as it units of Svedberg, which reflect rate of sedimentation of particle in ultracentrifugal sedimentation in ultracentrifuge, and velocity of sedimentation depend on size and depend on shape, and when it will be interact together, 50S and 30S, the result will be more compact than separatively. Now you can to see in 70S ribosome consist 23S ribosomal RNA and 60S ribosomal RNA main components plus 5S ribosomal RNA and 36 proteins associated with big subunit and 21 protein associated with small subunit. 80S ribosome consists 60S and 40S, more complex, a little, some more complex. 28S ribosomal RNA in big subunit and 18S ribosomal RNA in small subunit, plus 49 proteins in big subunit, 33 proteins in small subunit, plus 5S rRNA and plus 5.S rRNA. It's functional short RNAs. Really, RNA, ribosomal RNA provide it carcass of ribosome, and proteins provide main functional events in translation. And in ribosomes, in collected whole ribosome consists 4 sites for binding of components of translation. It site for binding of messenger RNA in small subunit, and 3 sites for binding of transfer RNA, complex transfer RNA, and amino acid. It P site, A site, P site peptidyl site, A site amino acid, aminoacyl site, and E site exit site for free tRNA after binding of amino acid with newborn growing polypeptide chain. Так, and ribosomes can be free or can associate with membranes of endoplasmatic reticulum. In this case, endoplasmatic reticulum can be smooth without ribosomes, and rough endoplasmatic reticulum with bound of ribosomes. It's important for next post-synthetic modifications of polypeptide chains. Ribosomes synthesize it's relatively complex processes of synthesis. Synthesis of ribosomes collection, association of ribosome take place in nucleolus, which present in nucleus, and it up to usually 3 nucleolus in nucleus, and it reflects complexity of synthesis, because ribosomal RNA synthesizes a single long chain of ribosomes in cluster of ribosomal RNAs, 45 you can see 45S ribosomal RNA precursor, which later will be digested on ribosomal RNA big and small subunits, plus proteins which necessary previously synthesized in cytoplasm and come in to nucleus, and proteins which will provide processing of ribosomal RNA. Altogether will be lead to separatively synthesized big and small subunits, which can be observed in electronic microscope, and at last big and small subunits will be exit from nucleus, and appearance of subunits in cytoplasm, and will be associated when it will be necessary for synthesis of polypeptide chain, collected only when will be associated with messenger RNA. Some details of synthesis. Messenger RNA translated in direction from 5 to 3' end. The same with synthesis of messenger RNA in transcription, by the way. And polypeptide chain synthesized from amino end to carboxy end. No, and ribosomal cycle, it initiation, elongation and termination. And you can see there it again I'll be repeat synthesis of bacterial prokaryotic messenger RNA, which can to consist multiple cistrons, will be beginning from Schneider-Lechner sequence, and its sequence localized in spacers between cistrons, all of which will consist start codon and Schneider-Lechner sequence. And independent binding of ribosomes, and beginning of synthesis when small subunit of ribosome will be interact with messenger RNA. For it necessary small subunit and soluble initiational factors, so-called factors which necessary only for initiation. For it, you're going to see it 3 main factors: initiational factor 1, 2, and 3. One factor first, it place of interaction with small subunit and preparing it to binding with messenger RNA. But for it at first will be initiational factor second by using of GTP bound of initiational tRNA. Initiational tRNA will be binding bound with P site of in future P site of small subunit, and it lead later to binding of messenger RNA, and Schneider-Lechner sequence will be specifically bound sterically with initiational factor 2, which will be interact with Schneider-Lechner, and in this case tRNA, initiational tRNA with methionine or formyl methionine in prokaryotes bound with messenger RNA. It's sterical physical interactions for correct interaction. First, initiational tRNA with correct initiational AUG. And initiational factor 3 provide check perfectness of interactions and when it will be complex format, it lead to binding of big subunit. Now you can see that initiation factors it proteins which bound small subunit. At first transfer RNA with methionine bound with small subunit of ribosome, then bound messenger RNA and join of big subunit. Next step, elongation. Again, after collection of joining of ribosome, necessary special also soluble factors which need only for elongation. It you can see it most rapid step of translation and orientation of rate of synthesis. It near up to 20 amino acids added per second. It's a moving of messenger RNA along ribosome up to 60 nucleotides per second. In eukaryotes it's less intensive, but also up to 2 amino acids per second will be associated. And for it necessary specific elongation factors will be provide elongation factors first OTU. U it unstable in increasing temperature. It protein. ITS stable. It really GTP and GTP mainly guanine nucleotide, which will be source of energy, and elongation factor G or elongation factor 2 for eukaryotes provide translocation of tRNA and messenger RNA. And for it you can see at first elongation factor TU or first bound with the GTP and with next tRNA. You know that number of tRNAs near 20, some more than 20, and it active tRNA with correspondent next amino acid will be bound with A site, free A site after binding of initiational tRNA. When it active complex of elongation factor will be interact with next codon, anticodon loop will be recognized next codon. The next step will be transpeptidase reaction associated with big subunit, proteins of big subunit ribosome will be lead to broken of ester bond between previous tRNA and forming of peptide bond with next amino acid, and next GTP associated elongation factor G or second factor for eukaryotes will be displaced complex from A site to P site, translocation of messenger RNA along ribosome or translocation of ribosome along messenger RNA as you want, and in E site will be a period free previous tRNA will be dissociated, and in P site will be a period next tRNA with associated polypeptide chain. Polypeptide chain will be synthesized in free amino acid, will be from free amino acid end. And again, next elongation factor 1 association with next triplet, translocation, and more. And each step, on each step, necessary GTP, 2 molecules of GTP, one for interaction with the next transfer RNA, and next for translocation of ribosome. You can to see that elongation factor, which will be translocate messenger RNA and ribosome, you can to see has similarity with amino acyl tRNA synthetase, but affinity to P site when it bound will be more for ribosome than tRNA. And now you can to see the hot spot of synthesis. Now it P site of ribosome amino acid site and exit site. Very important for it, 5S RNA, which will be provide effectiveness of interactions, and take place movement of polypeptide chain from amino acid, from amino end, which will be go across channel in big subunit, and movement of messenger RNA from 5' end to 3' end. And at last will be a period of free A site with appearance of stop codon. In this case, will be interaction with terminal factors or releasing factors. It also 3 factors, one of which will be recognized at its place. For it again necessary GTP. You can to see GTP is source of energy for synthesis of polypeptide chain for translation. In contrast with ATP in metabolic pathways, it leads to absence of competition between protein synthesis and metabolism for source of energy. And at last terminal factor will be digest last ester bond between polypeptide chain and last tRNA, and it leads to dissociation of messenger RNA and ribosome. Now, by the way, you can to see one messenger RNA can bound multiple ribosomes. It depends on affinity of 5' end of polypeptide chain of 5' end of messenger RNA to a small subunit of ribosomes, and it leads to forming of polysome. Now some interesting fact about possibility of determination of ancient temperature on ancient planet, on our planet, when bioinformatics lead to possible calculation of ancient polynucleotide sequence in elongation factor 2. It possible to calculate by comparison of sequence of nucleotides in elongation factor in different organisms in different steps of evolutionary elongated distance one to other, and the calculation of it ancient nucleotide sequence and later synthesis of amino acid sequence from it onset polynucleotide system sequence and determination of optimal temperature of working led to temperature of onset temperature of optimal working near 60-65 degrees. In contrast with optimal temperature of elongation factor for organism which live now at E. coli and at onset temperature. Three independent calculations led to the same result, near 60-65 degrees. You see it, our planet gradually decreased temperature, and near 3 billion years ago temperature was some more. Now look at last universal common ancestor. It proposed total precursor for all living organisms in our planet. And you see minimum for ATP necessary for association each amino acid. And multiple steps of regulation of translation possible. It changing of activity of participants of translation. It changing of rate of initiation. It number of messenger RNAs and the number of ribosomes. Now again, you can see, I will repeat, the structure of messenger RNA on eukaryotes. It site for binding with ribosome, which provide frequency and affinity to ribosomes, frequency of interactions. It coding part and untranslated 3' end, polyadenyl tail, plus factors which provide stability, and next information about different variants of participation of polypeptide chain in different parts of cell space. And important factor, for example, for activity of synthesis, elongation factors. Elongation factor 2, by the way, it used for calculation of temperature, onset temperature of our planet. It so-called housekeeping gene coded it factor, and phosphorylated and non-phosphorylated steps of it lead to changing of its activity. For example, phosphorylation lead to stop of synthesis because in this case necessary specific conditions for cells and stop of protein synthesis. And initiation factors also can be phosphorylated and then not phosphorylate. It also will be influence on velocity of synthesis. And on translation, possible changing activity by specific factors which will be destroyed of synthesis. It reflects serious situation of life of prokaryotic world, and you can see there, for example, interaction with 30S subunit of ribosome will be lead to destroying of correct codon-anticodon interaction of amino acyl tRNAs and growth and synthesis. It mechanism action of streptomycin and pyrimycin. Pyrimycin correspond to 3' end of amino acyl tRNA, and it will be competition for A site between tRNAs and A site. Puramycin it inhibit of first step of translation. Tetracyclines also interact with 30S ribosome subunit, prevent binding with next amino acyl tRNA. Cyclines, tetracycline or monocycline, next generation of antibiotics. Macrolides, it interacts with 50S subunit of ribosome, prevent transpeptidase activity. Transpeptidase activity is function of 50S ribosome. No stop of synthesis and, for example, erythromycin. Chloramphenicol prevent elongation because again it inhibition of peptidyltransferase activity. Bound with 23S ribosome, prevent forming of the peptide bond. Так, you see it, chloramphenicol, triamphenicol. All of it influence on synthesis of bacterial prokaryotic synthesis. It lead to effect for eukaryotic organism against prokaryotic. Fusidic acid prevent exit factor of ribosome, again lead to blocking of elongation. It was antibiotics for prokaryotes, but antibiotics for eukaryotes also take place. Multiple antibiotics. It used for anti-oncogenic anti-proliferational factors for tumor cells. You can to see different steps of translation can be inhibited by correspondent antibiotics. No, oncology will be studied more detailedly. And inhibitors can be mechanism of action can be influence for eukaryotic. For example, diphtheria toxin inhibit elongation factor 2 by ADP rebodylation with modified amino acid histidine, diftamid, so called, important for function of elongation factor 2. It's a toxin of diphtheria, very important for it. Cycloheximide inhibit elongation chain for pro and eukaryotic cells together, prevent action of peptidyltransferase. All of it very important for synthesis. Next problem, after appearance of polypeptide chains, each polypeptide chain necessary will be distributed in functional site. And it appeared problem because multiple synthesis can lead to dysfunction if proteins will be work in non-usual, not oven place. But all of it effectively distribute because at the each polypeptide chain present specific so-called sorting signal. And all of it important for distribution of cells. Now you can to see that our inner space of cell functional divide on two volumes. It's cytoplasm and endoplasmatic reticulum. Nucleus is part of cytoplasm because for transport of substance from nucleus and from cytoplasm not necessary pass across membrane. But if substance must be secreted from cells, it must go across membrane, and it's big problem for synthesis. For next post-synthetic modifications, because membrane is lipid inner layer, and it lead to necessary of unfold of polypeptide chain and refold after pass across membrane. It's important for Golgi apparat, for lysosomes, for secretion. Golgi apparat is system of membranes which will be provided effectively. Any protein synthesized in cytoplasm, any protein synthesized in endoplasmatic reticulum. And again, rough and smooth endoplasmatic reticulum, rough is bound with ribosomes. And its factor important for next transformations of polypeptide chains. Now, in prokaryotic ribosomes, synthesis takes place in cytosol, but for eukaryotics, it can be synthesis or in cytosol or in endoplasmatic reticulum. It depends on specific sequence which will be present in polypeptide chain at the beginning on amino end of polypeptide chain. When polypeptide chain will be appeared in extra ribosomal space, it will be consist specific signal sequence which indicate about its appearance. It usually hydrophobic and positively charged groups of amino acids. Special signal sequence. It will be recognized by a single signal recognizing particle, complex of proteins, which will be bound of signal sequence and provide interaction with membrane associated complex in endoplasmatic reticulum membrane. And later, now you can to see polypeptide chain will be secreted to bound with membrane, and the polypeptide chain will be secreted to cistern of endoplasmatic reticulum, volumes of endoplasmatic reticulum, which later will be associated with Golgi apparat. And the polypeptide chain will be appeared in extra endoplasmatic reticulum cisterns, lumen, and the enzymes associated with which will be associated with endoplasmatic reticulum membrane will be modified next polypeptide chain. Next, it folding of polypeptide chain. When we told about structure of enzymes, that primary structure of polypeptide chains it's main factor for its folding. But in cells, when concentration of proteins are relatively high, it can lead to non-planned random interactions between newborn polypeptide chain and other proteins. For prevent of it, We have special proteins which call chaperones. If polypeptide chain will be folded incorrectly, it will be lead to quick digestion of polypeptide chain by proteolytic system. Its proteasome will be effectively digested. It will be quickly labeled by labels, for example ubiquitin, and it will be digested, and in this case cell can destroy incorrectly bound, incorrectly folded proteins. If proteins will be accumulated in cells, if non-digested, wrong configuration of proteins will be accumulated cells, it so-called prions, it will be digested, it will be lead to cell activation of apoptosis. Cell will be dead. And for correct folding, necessary special proteins which called chaperones. Other name it head shock proteins. For example, you can to see one of chaperones. It's cylinder of protein cylinder which will be associated with newborn polypeptide chain and provide correct folding of it. Chaperones in cells can be divided on three major classes, main classes. It other name chaperones, it head shock proteins, and molecular weight of its proteins lead to HSP 70, 60, 90. All of it are important for effective folding, facilitate of effective folding. Now you can to see standard signal sequence which will be upstream of proteins which must be secreted. Hydrophobic and positively charged groups. It will be digested later by nuclease, and active part will be folded and excreted from the cells. And next, for synthetic modifications. It proteolytic cleavage, yes. We told that all amino acids beginning, all polypeptide chain synthesized from methionine, but methionine later will be digested. Next, forming of disulfide bonds, and modification by special groups, glycosylation, acetylation if necessary. Phosphorylation, important for regulation of activity. And amino acid modifications, for example, hydroxylation of proline, for example, methylation of lysine, and multiple different variants. For example, modification of polypeptide chains for synthetic modification. It collagen. Collagen is protein which consists of 3 polypeptide chains associated one to other. Association between chains non-covalent hydrophobic interactions, and primary structure of collagen polypeptide chains. It repeated motif glycine, proline. Sometimes alanine, sometimes leucine, sometimes lysine. But for effective interactions between chains, necessary for synthetic modification of proline to hydroxyproline. If hydroxylation will be imperfect, in this case, temperature of dissociation of chains will be low, and for hydroxylation necessary complex with participation of vitamin C, ascorbic acid. If will be inefficiency of ascorbic acid, hypovitaminosis for example, in this case hydroxylation of proline post-synthetic modification will be imperfect, and melting temperature of collagen dissociation will be decreased to temperature of human body. It will be lead to forming of clinical picture of scurvy. And the regulation of transcription. Return to organization of genome. We told that each DNA in prokaryotes can be organized in operonal type. Near half of all genes organized in operonal type. Operon is part of DNA which consists promoter, part binding of RNA polymerase. Between promoter and operator will be short polynucleotide chain, near 10 nucleotides, which call operator, and multiple structural genes, maybe 2 and more. Each gene cistrons coded one polypeptide chain. It different proteins, different enzymes, but total properties of all it enzymes, it participation in the same metabolic pathway that necessary for synthesis of any amino acid, for example tryptophan, operon, or breakdown of any source of energy, for example lac, operon. And transcription of it will be lead to synthesis of long messenger RNA, which will be consist multiple cistrons. Each of it will be coded correspondent proteins. And theory of regulation described at first Jacob Monod theory. You can see there. Now you can see operon, functional unit of DNA, which consists cluster of genes by control of single promoter. And operons can be divided on two groups. It inducible, which will be coded enzymes for metabolism of metabolic source of energy, for example lac operon, will be normally off, but will be switching when it necessary. And repressive, normally on, but off if necessary. All of it function depend on promoter region. Promoter region consists site specific sequence which will be recognized by RNA polymerase. RNA polymerase in prokaryotes consists sigma factor, which will be recognized promoter region. Now you can see different variants of promoter. It TA, different variants of so-called Pribnov box. In eukaryotes, TA box. It conservative sequence and conservative sequence -35. Between them maybe anywhere. And later AUG will be beginning some later. And and other parts of RNA polymerase core enzyme will be recognize sigma subunit associated with promoter. And now you're going to see promoter, and between promoter and coding path present short path operator. When messenger RNA bound with promoter, it can go ahead, but operator, state of operator can influence on its activity. And if operator will be bound with repressor protein, in this case synthesis will be absent. Operator it short part of DNA which can bound with molecule of repressor. Repressor can be active or can be inactive. If repressor active, it bound to operator and it prevent of transcription. Repressor is protein which coded by special gene which will be not part of operon. Operon consists promoter, operator, and structural genes, cistrons. Regulative gene or I gene, it's gene which consists information about repressor protein. And repressor again can be active and can be inactive. When, for example, E. coli live in environment when present glucose and absent other source of energy, not necessarily synthesized anywhere, but glucose will be finished but appeared, for example, lactose. In this case, if in genome present lac-operon, lac-operon will be before inhibit, because repressor will be active and bound with operon, operator. Appearance of lactose lead to binding with repressor. Repressor will be modified and dissociate. In this case, inactive. In this case, RNA polymerase will be synthesized polycistron messenger RNA. Cistrons, it coded of enzymes, it beta-galactosidase, permease, and acetylase, all of it necessary for metabolism of lactose. Lactose present, operon is active. Lactose absent, operon is blocked. But present specific place which can regulate activity of promoter. Repressor will be active, and promoter in inducible operons usually low active, but when in cells will be low concentration of ATP, a low concentration of glucose, in this case it will be lead to forming of cyclic AMP. Cyclic AMP is strong mediator for intracellular stress factor. It will be interact with cyclic AMP binding proteins which will be recognized and bound in promoter region, and it lead to increasing of affinity of promoter to RNA polymerase. Weak promoter will be transformed to strong promoter, and it will be additional factor for increasing of activity of it operon. Repressible operons. Operons, it usually operons which will be coded enzymes for synthesis of any, for example, amino acids or any other useful substances. it's histidine or tryptophan operons. In this case, mechanism is the same. RNA polymerase bound with repressor, and if repressor RNA polymerase bound with promoter, and if repressor will be bound with operator, in this case, transcription will be absent. Free operator will be lead to activation of synthesis. But now repressor synthesized, but it inactive, and operon will be active. But appearance of any substance, target substance, for example, histidine with correspondent repressor, will be lead to its activation. Activation bound with operon, and transcription will be stopped. And again, like lac-operon will be mediated by will be modulated by cyclic AMP. Repressible operons also can regulate, but additional regulation, but it will be regulated by translation. For example, now you can see his operon, which consists minimum 9 cistrons. But regulative part of it, now you can see enzymes which can transformate synthesis of histidine from phosphoribasyl pyrophosphate. Phosphoribasyl pyrophosphate, substance which will be appeared from pathway of pentosophosphate pathway. And any gradual transformations lead to synthesis of histidine. But upstream of messenger RNA will be present specific place which will be has 4 parts of messenger RNA, which will be G, D, C, and E part. Upstream of coding sequence. Ribosome will be go across path which will be increase multiple triplets of histidine. Its additional factor for checking is histidine really low, and if histidine, concentration of histidine will be influence on velocity of moving of ribosome along its path L path. This is histidine triplets. You see CAC, CAU. Так. And variant of messenger RNA D and C, one to other, consist variant sequence C can folded interact with D sequence with forming of terminal loop. Terminal loop it place for dissociation of ribosome independently of sequence. And if D will be interact with G, in this case will be non-terminal loop. Situation is next. when concentration of histidine in environment relatively low but not too low, in this case ribosome relatively quickly pass across L path and go to the G path. In this case D sequence will be forming with the C sequence of the same messenger RNA. Forming of the terminal loop and ribosome will be dissociated. Stop of synthesis, stop of translation. But when histidine concentration really low, in this case ribosome will be very slowly moved along L path. In this case G will be sequence will be create D interact with G non-terminal loop, and C will be participate in other conformational changing of ribosomal messenger RNA. In this case non-terminal loop and all enzymes of hisapiron will be synthesized and histidine will be synthesis of histidine will be take place. It so-called attenuator system. Also next regulation of gene expression by growth vectors. For example, it growth vectors hormones which will be activate synthesis of polypeptide chain by corresponding genes, but between growth factor which will be recognized by a receptor and intracellular and the intracellular chain of events will be finished by transcriptional factors which will be inactive or active. Phosphorylation, dephosphorylation of it lead to it activation, inactivation. And the chain of events when growth factor interact with the receptor, it will be lead to it phosphorylation. Phosphorylation or receptor will be lead to next steps of phosphorylation of intracellular protein kinases, and protein kinases will be phosphorylate factor of transcription, and it lead to synthesis of group of genes which important for cell proliferation and differentiation. Again, one of factors, it parts of DNA which will be localized far upstream of promoter region. It so-called enhancers. Enhancers, it place of DNA which will be specifically recognized by specific proteins, activators of transcription. Without its factors, active transcription will be impossible. Now it example when it enhancers which will be recognized by complex of steroid hormones and receptors. Now steroid hormones can pass across membrane to cytoplasm, recognized by receptors. Receptors was before inactive, but interaction with hormone which will be lead to its activation. Complex will be interact with enhancer. It enhancers called hormone responsive elements, and it will be lead to activation of RNA polymerase and activation synthesis of corresponding polypeptides. Sometimes activators factors of activation will be products of other regulative genes, and it products will be activate synthesis of other regulative enzymes, and totally it will be appeared multiple complex nets of regulation. Sometimes it comparison with the regulation of neurons in our brains. When neurons different levels regulate other regulative neurons. The same there. When signal from environment will be activate synthesis of regulative genes, and later it will be lead to activation next genes next order, and at last will be lead to synthesis of multiple proteins which necessary for any specific synthesis, for example, now chondrogenesis. Destroying of it net will be lead to multiple problems with now chondrogenesis, for example, rheumatoid arthritis, or any other events in cells, including oncotransformations. Also, regulation of protein synthesis is basis for a long-term memory. For example, intraneuronal interactions lead to more effective forming of interneuronal nets, it's basis of our memory, and repeating of expressions lead to storage of it system, short time memory, it only facilitation of it interneuronal interactions, but long time memory it depends on synthesis of specific proteins, which or increasing number of mediator which will be synthesized, or increasing of number of receptors of it mediator, and all of it will be lead to more effective interactions between neurons and more effective reproduction of its impressions if necessary. Mutations. Mutations is the usual natural changing reasons lead to changing of sequence of nucleotides. It can be changing of nucleotides, gene mutations, substitution, deletion, addition. It result of maybe a result of modification of nucleotides. For example, spontaneous oxidation can lead to changing of amino group to oxygen, and it lead to changing of, for example, cytosine to uracil. But if it will be masked by methyl groups, it can be transformation of methyl cytosine to thymine. Thymine is normal usual nucleotides. Multiple depurination also. Some thousands of purine bases can easily spontaneously hydrolysate, and for it necessary provide of it reparation. Also alkylation agents. All of it lead to changing of configuration, ultraviolet also. It lead to necessity of reparation, and in our cells we have effective system for DNA reparation. It may be reparation by if mutations take place in one strand. For it, reparation provided by, for example, base excision, or excision big or more or less intensive excision of reparation, and specific signal which will be regulate of events of modification, especially very dangerous for mutation it steps of replication of DNA and transcription, because in this case will be appearance single-strand DNA and its effective strong dangerousity of destroying. And if will be take place break of both strands, it's very dangerous, because reparation possible or by homologic recombination, when in cell present second set of chromosomes, homologic chromosomes. It take place in S or G2 phases of cell cycle, or if it will be G1 phase of cell cycle. In this case, it will be realized across non-homologic end joining with missing of genetic material between breaks. It's more dangerous. Well, multiple mutations lead to changing of genetic material in the old cells. We haven't two cells with the same genetic materials. All cells are after dividing will be has modification of genes, and it lead to good material base of evolution, but evolution in organisms with origin from one cells, for example humans, it can be different, can be difficult, dangerous. And in our genome we have senseless sequences, in contrast with prokaryotic. In prokaryotes, senseless sequence relatively low. It usually elements of defense against mutations, against fags of and other elements of immunity of prokaryotes. In humans and other eukaryotes, we have a lot of senseless sequences. Sometimes it's called cemetery of promoters. And now you can to see elements of human genome. Only some persons of genome it coded proteins, functional units, but mainly it transposon based or heterochromatin or other non-conservative sequences. In contrast with mitochondrial genome, there is only little part of genome senseless, but mainly all of it very important for coding. Now you can to see genome organization. We have 3 billion nucleotides, 3.2, and near 10% of it coding DNA. Other, it different variants. It's pseudogenes, genes without active promoters, fragments of genes, introns, and other. And mitochondrial genome, where you can to see 2 RNase genes, ribosomal RNase genes, 22 transfer RNA genes, and polypeptide coding genes. All of it very conservative. Now you can to see again introns and non-coding sequence, but maybe functional active microRNA in the other, up to 30%. A long interspecies non-coding sequence and a short interspecies non-coding sequences, retroviruses and unknown sequences. You're going to see all of it important for the existence in our genome. And sequences and mutations has different entrance important for diseases. For example, you can to see examples when any diseases strongly associate with mutations, any node. No, for example, very high level of importance of mutations for single monogenetic diseases. For example, cystic fibrosis, phenylketonuria, or Huntington disease. Any case, diseases depend on genetic predispositions, but also influence on environment. And low genetic influence, for example, car accident and other, you're going to see. Infections more depend on environment, but also genetic entrance, also important. Now we're going to see monogenic diseases, polygenic diseases, predispositions, and environment. All of it lead to complex distribution of importance of genetic material for development of disease. One of it, sickle cell anemia. Mutation on one gene, one mutation lead to development of disease. Result, appearance of mutation of hemoglobin, so called sickle cell. Substitution of valine instead of glutamine in position 6 in beta chain. It result of mutation instead of GTC will be coding G normal glutamate, GUC, but if instead of it will be CAC, it will be lead changing to valine and lead to more hydrophobic nature of hemoglobin and sedimentation of hemoglobin in strands with reduced hemoglobin. In oxidized state, it normally, but in venous blood, it will be the deformation of erythrocytes. Distribution of sickle cell anemia correspond to distribution of malaria, because malaria plasmodium cannot effectively develop in sickle cell anemia erythrocytes. The name of next variant is thalassemia. Thalassemia is result of destroying of correspondence between alpha and beta chains of hemoglobin. Normally, we can synthesize hemoglobin A adult. Synthesis of 2 alpha chains and 2 beta chains of globins. And alpha chains, we have 2 genes of alpha chains and 1 copy of beta chains, but synthesis must to be by the same concentrations. Now you can to see beta chains coded by chromosome 11, alpha chains chromosome 16. Altogether necessary associate in hemoglobin, normal hemoglobin. Normal hemoglobin consists alpha 2, beta 2, beta 2 chains. But if synthesize will be synthesis more alpha chains than beta, it will be lead to destroying of normal functions. It will be alpha thalassemia. If more will be beta chains, in this case will be beta thalassemia. We can synthesize in fetal a little in embryonal development fetal hemoglobin alpha 2 gamma 2 and a little synthesize hemoglobin A2 alpha 2 delta 2. Delta is variant of synthesis with increasing affinity to oxygen. Now you can to see correspondence between synthesis of alpha globins less 2 times and beta globins with a more effective synthesis, and destroying of it lead to thalassemia. Analysis of primary structure of globins lead to possible to appearance of ancestral globin, which will be appeared near 8 million years ago. Enzyme disorders. Mutations in genes of correspondent enzymes lead to destroying of normal metabolic pathways. For example, glucose 6-phosphate dehydrogenase deficiency. It most frequent distributed mutation in human population, all of it associated with regions of humid, hot climate in Africa and Eastern Asia, with a high distribution of malaria, events of malaria. Glucose 6-phosphate dehydrogenase, an enzyme which will be transformate glucose 6-phosphate to glucolactone and 6-phosphogluconate. It pathway lead to reducing of NAD phosphate, and it important for anabolic reactions and for support of reducive conditions in cells. And more oxidative destroying of it pathway lead to more oxidative conditions in cells, including erythrocytes. In this case, erythrocytes more stable. Its body more stable for malaria, but erythrocytes less stable, and any events in oxidative stress lead to hemolysis of erythrocytes. Next, we told that phenylketonuria, it mutations in gene of phenylalanine hydroxylase, which localize in chromosome 12. In this case, it lead to accumulation of phenylalanine and phenylpyruvate in blood, and it lead to dysfunction of maturation of central nervous system and oligophrenia. Some lesson ago we talked about it when told about metabolism of phenylalanine. Maple syrup urine disease. It also dysfunction of alpha ketoglutarate, alpha keto acid derivatives of hydrophobic branched amino acids. It leucine, isoleucine, and valine. Result will be accumulation of correspondent alpha keto acids And normal metabolism, it next oxidative decarboxylation. When it alpha keta acid decarboxylation will be destroyed, it will be lead to accumulation of alpha keta derivatives, alpha keta acid derivatives, and dysfunction of central nervous system. Well, one of symptoms of it will be maple syrup odor. But of course more important is oligophrenia and the dysfunction of it. Albinism. Now we can to see again block of metabolism of phenylalanine tyrosine dysfunction. Now you can to see the synthesis of melanin pigments and lead to changing of intensity of blood skin color. We told that in equatorial zone necessary to have dark skin and a high level of melanin for defense against mutagenic ultraviolet. For high altitude living humans necessary to have a light white skin. For absorption of ultraviolet necessary for synthesis of vitamin D, vitamin D3. Next, lysosomal storage diseases. It dysfunction of enzymes which digest oligoglycosidic chains. For example, synthesis of glycolipids and sphingolipids. It different groups of diseases. For example, Tay-Sachs disease or other multiple. All of it lead to accumulation of non-digested oligosaccharides in lysosomes, and at last cells will be died. Serious neurodegenerative diseases. No, and diseases, genetic influence on diseases can be described as polygenic and multifactorial inheritance. When more than one gene mutation and genetic variants of more than one gene lead to more predisposition to any diseases. When environmental conditions lead to more influence on development of it. Now we can to see polymorphism, genetic polymorphism, main factor for existence of population, but also reasons for development of multifactorial situations. Next, oncogenesis. Mutations in receptors of regulation factors, mutations in intracellular chains of events which will lead to activation or inactivation of factors of proliferation, lead to non-controlled development of proliferation of cells. For example, you can to see multiplicity of it lead to multiplicity of variants of oncotransformations. Mutations can lead to point mutations, deletions or insertion to DNA and correspondent proteins, hyperactivation of regulative proteins. It may be amplification of genes, super expression of correspondent genes, and chromosomal, for example, rearrangement lead to dysfunction of synthesis of correspondent genes. And the mutations, anti-oncogene genes, tumor suppressor genes, destroying of activation of apoptosis or gene reparation and lead to dysfunction of it. One of so-called guard of genome is protein P53. It will be activation by multiple factors, and its activation lead to arrest of cell cycle. And if reparation will be imperfect or impossible, too intensive metabolism, it will be lead to activation of apoptotic transformations. Now we'll be finished. Thank you very much. Goodbye.